In the innermost cortex, it is now known as bacteroid. So now they will sit in their drawing room, which is the innermost cortex, and now they will start their discussion. Then the bacteria will ask the plant that since I am coming after a long period of 5 years, I have brought some special sweets for you. So the bacteria has brought some special sweets for the plant body. And what is the name of that sweet? The name of that sweet is cytokinin. And the plant likes it very much, cytokinin. Then when bacteria will ask the plant, what you have brought for me? Then the plant will immediately secrete auxin. That means bacteria is secreting cytokinin and plant is secreting auxin. Both are plant hormones. And when these two hormones will be present together, what will happen? You know it. Now there will be a rapid cell division. There will be rapid cell division in the inner cortical cells. And the cells will divide so rapidly that sometimes the karyokinesis may not be followed by cytokinesis. So some of the cells also become polyploids. And as a result of this rapid cell division, there is a proliferation of cells in the innermost cortex. Here the cells will be proliferated because of the large division. So now this will come out in the form of a nodule. And this is known as a root nodule. And this root nodule has a direct connection with xylem and phloem, that is vascular tissues. And this nodule formation is again favored in presence of two elements. One is molybdenum and the other is sulfur. That means the nodule formation is favored by these two elements, and it is due to the presence of these two plant hormones, cytokinin secreted by bacteria and auxin secreted by the plant. So now the root nodule comes out, and when the cytoplasm of the root nodule is analyzed, it is found that inside the root nodule there is one pink pigment. There is one pink pigment, and this pink pigment is known as leghaemoglobin. So why it is known as leghaemoglobin? Because it is just like hemoglobin present in vertebrates. Why leghaemoglobin? Because this hemoglobin is present in leguminous roots. So that is why it is leghaemoglobin. Now this leghaemoglobin, listen very carefully. This leghaemoglobin is not present in bacteria. It was also not present in plant body, but it is present in root nodules. Even it is not present in other cells. That means the formation of leghaemoglobin is the symbiotic product of bacteria and the plant. This is the product of symbiosis, because it is neither present in bacteria nor in plant, it is also not present in the cells outside the nodule. It is present only in the root node. That means it is the product of the symbiosis. Now what is the main role played by this hemoglobin? This main role is it functions as oxygen scavenger. Oxygen scavenger. That means when a nitrogen fixation takes place, usually the nitrogenase enzyme, or we can tell it is a dinitrogenase enzyme. This dinitrogen enzyme is very, very sensitive. It is very sensitive to oxygen. And without this enzyme, nitrogen cannot be fixed. So when there will be nitrogen fixation, usually, as already I have told you, that rhizobium is aerobic bacteria. Why it is aerobic bacteria? Because for nitrogen fixation it requires a lot of energy, and this energy cannot be provided by anaerobic bacteria. So that is why the aerobic produces sufficient amount of ATP. But when this nitrogen will be fixed, it has to be fixed under anaerobic condition. So oxygen is available, how anaerobic condition will be provided? For that reason, the leghaemoglobin, which is produced as a result of symbiosis, that removes the oxygen from the site of nitrogen fixation, so that in anaerobic condition, nitrogen will be converted to ammonia. This is again very important thing. That means rhizobium is aerobic, but nitrogen fixation takes place in anaerobic condition. Rhizobium is saprophytic, but when it comes in contact with the roots of leguminous plants, it becomes symbiotic. These are very two important things. So this is the main function of this rhizobium, that is the pink pigment. Now another very important thing is all these things that are taking place, it is all are genetically controlled. That means the symbiosis, the product formation of leghaemoglobin, all these are under genetic control. And usually there are a cluster of genes. There is cluster of genes which help in the formation of the root nodules and formation of leghaemoglobin and the nitrogen fixation. So here in the plant, there is one gene that is known as NOD gene. And in case of bacteria, that is rhizobium, there is a NOD gene, then a NEET gene, and another is a FIX gene. There are 3 genes. So in bacteria, 3 genes, NOD, NEET and FIX, and in the plant there is one gene, that is NOD gene. So it is the combination of these 4 genes, or a cluster of these 4 genes, which take part in the formation of root nodule and the leghaemoglobin and the nitrogen fixation. So this is the method how the root nodule is formed, and within this root nodule, how the nitrogen will be now fixed to ammonium compounds. Now let us discuss that part, the mechanism of nitrogen fixation. So already I have told you that nitrogen fixation requires a lot of energy, and the mechanism is usually the nitrogen, it combines with 8 electrons, 8 protons, and 16 molecules of ATP, and in presence of the enzyme, the dinitrogenase. And again, this is dinitrogen enzyme. In presence of this dinitrogenase, now what will happen? It will give rise to 2 molecules of ammonia. Then it will give rise to 1 molecule of hydrogen. Then also it will give rise to 16 molecules of ADP and 16 molecules of the inorganic phosphate. So that means for production of 2 molecules of ammonia, it is requiring 16 molecules of ATP. That means for each ammonia molecule, 8 ATP molecules are required. So much of energy. How can it be provided by anaerobic form of bacterial rhizobium? That is why it is always aerobic, because it requires sufficient amount of energy. So if we write this reaction as 1, and in another reaction we write as the 2 molecules of ammonia will combine with this hydrogen, and it will give rise to 2 molecules of ammonium ions, and 2 electrons are liberated. Okay. Number 1, number 2. If we will combine both these reactions, what will happen? We will see that nitrogen combines with the 6 electrons, 8 protons, and 16 ATP. 16 ATP, and it gives rise to 2 molecules of ammonium ions, then 16 ADP and 16 inorganic phosphate. So this equation is again very important from examination point of view. It can be direct asked as a question. So, but when ammonium ion should be formed, then this is the equation. For ammonia formation, this is the equation. So in this way, the nitrogen is fixed. This is the mechanism. And this takes place only in the presence of this enzyme dinitrogenase. Now this is dinitrogenase. How the whole thing functions, let us discuss also diagrammatically. Now this is the enzyme dinitrogenase and these are the active sites. So in the next step, what is happening? Now it will combine with the 2 molecules of nitrogen. That means now nitrogen will come to this place. So when the nitrogen will come to this place, now it will look like this. That means now here will be nitrogen and here will be nitrogen. Now nitrogen is combined together in the active site. In the next step, there will be the hydrogen will come. So in the next step, this is nitrogen, this is the nitrogen. Now 2 molecules of hydrogen. When the hydrogen will go to this place, it will combine with it will combine with 1 hydrogen and it will also combine with another hydrogen. That means it is NH. And in the next step, another again 2 hydrogen atoms will go. So now this will be nitrogen, and it will be NH2 and NH2. And in the last one, again 2 hydrogen ions will move. Again 2 hydrogen, and now it will be N, this will also be N, and it will be attached with 3 hydrogens, and this will also be attached with 3 hydrogens. So now this is NH3 and NH3, and then the finally what will happen, these 2 ammonia molecules will be liberated, and now the enzyme is free. Now the enzyme is free. So in this way, at the active site of this enzyme, the nitrogen combines, and in step by step, the hydrogen combine together. And for each step, it requires sufficient amount of energy, and that energy usually comes from a respiration. And for that purpose, the rhizobium is aerobic. So this is the total mechanism of nodule formation and mechanism of this nitrogen fixation. So now we have seen that inside the soil, the atmospheric nitrogen is fixed in the form of ammonium compounds. And after this, what is happening, that we'll discuss in the next video. But let us discuss some questions. So one question, leghaemoglobin is a product of HOS bacteria B choice plant, C choice symbiosis, D choice none of the above. This one question. The second question, nitrogen fixation takes place under A choice aerobic condition, under condition of aerobic, anaerobic, C choice fast anaerobic, and then aerobic, and D choice none of the above choices. So these are the standard questions for all national type of examinations. Thank you very much for watching it. I hope now you have a very clear idea how the nodule is formed, how nitrogen is fixed, and what is the nature of rhizome bacteria. God bless you. Have a good day.